All right. Hello, everybody. StreamYard's telling me we're live, but StreamYard occasionally lies, so I'm going to wait till I hear it from YouTube. Once YouTube tells me we're live, then I'll actually get going. James, I don't know if you've ever done that, but I've done that where I'm talking and you can't hear anything on YouTube, so I always wait a minute just to make sure. And I can, in fact, hear myself, so that's great. Okay. Hello, everybody. We are now officially starting. I'm Dan. This is Creationists. We're here with Dr. James Carter, and we are going to talk about the Lenski long-term evolution experiment. So I'm going to let Dr. Carter introduce himself, and then we're going to go right into a brief introduction to the experiment from Dr. Carter, and then we'll talk about the Lenski long-term evolution experiment because it is just a fantastic. It's one of the all-time great experiments in evolutionary biology, and I'm super excited to be talking about it. So with that, I am going to turn it over to Dr. Carter for a few minutes, and then we'll talk about the Lenski experiment.
That's so weird. I'm getting a double, like a relay that's like 5 seconds behind, and I don't have any other slides open. So I guess when I'm talking, it won't be a problem. I just hope that, oh, now I'm hearing myself like 10 seconds later.
I'm trying to think. I don't have anything playing out loud. Yeah, I don't have anything out loud.
Okay.
I'm not sure where that would be coming from.
That.
Let's see. Yeah, I am. Is anybody listening have an echo, or is it just for Dr. Carter?
Well,
it's possible you have a window open somewhere.
I didn't even open YouTube to make sure that didn't happen. Maybe. Oh, you know what? Huh. I do have one of the tabs.
There it is.
There. Okay. Sorry, I had that up from yesterday. Much better. Okay. Yes. Sorry about that. I am James Carter from Southern California. I teach at Loma Linda University, where I got my PhD, and I teach a bunch of science classes. And Dan invited me onto his channel, I guess quite a while back, and we were trying to decide on a topic. I'm fairly new to the YouTube community. Last couple months, I've been giving some talks here and there on abiogenesis on a few of our friends' channels that we both visit. And so I thought, well, I need to kind of get out of my comfort zone and go on some topics that maybe I'm not as familiar with. Hopefully, I can learn some things from Dr. Dan, and so I thought, what a better way to learn something than to teach it. So I've been familiar with Richard Lenski's experiment that's been going on for quite a while now, but I wanted to get caught up on what the latest information was. So I will do a brief overview of the research for those of you that aren't familiar with it, and then we can take some time to fill in any gaps. And then I think the most interesting part, we're going to spend the majority of our time discussing the implications of this experiment. So let's see if the slide advances here. Okay. So this is an example of a long-term evolution experiment. And, rather than just rely on the fossil record and trying to establish phylogenies, this is some real laboratory work here where a strain of E. coli has been grown in the laboratory at Michigan State University. Quite a few people have worked in Dr. Lenski's lab over the years. In fact, they now have 265 publications. I guess there's another one in print that I couldn't get access to. So I guess that's going up, even though the experiment's on pause.
Can I just say, funny connection with the Lenski Lab. Lenski is my academic great-grandfather, if you trace back thesis advisors.
Oh, wow.
So I am tangentially in the family tree of Lenski Lab alum. So my thesis advisor was in the Lenski Lab for his. My thesis advisor's thesis advisor was in the Lenski Lab for his PhD. There's a fun fact. It's cool. A big family tree at this point.
Yeah. So the overview I'm using, since there's so many publications to go through, or stuff I've grabbed from his papers, as well as a review and critique. A couple years old. I have some newer references in the presentation as well, but you can download this paper at dismantledevolution.com under the resources section. And I'm sure many of you who are familiar with the work that's going on will recognize John Sanford, the author of Genetic Entropy. I'm familiar with him because I did some plant genetic engineering in my graduate research, and my mentor, the principal investigator, was a very he knew him well because he was my mentor, William Langridge, did a lot of research in transgenic plant vaccines, edible vaccines for various autoimmune and infectious diseases. We use a different technique than the gene gun that John Sanford invented, but yeah, he's a great guy. I met him a couple times when he came and spoke at our university, Loma Linda University, a couple years ago. So let's go over the basic experimental design. Started it way back in 1988. So imagine trying to be a graduate student and get some results in a timely matter. Obviously, they're doing really well with all those publications, but that is a really long experiment as far as experiments go. 33 years ago, just over 33 years ago. They started with 12 separate cultures from initially genetically identical strain of E. coli. So they grew them in these little culture flasks that have all the bare essential nutrients for the E. coli to grow, and then they mechanically swirl it around. It's grown in oxic conditions, so they're E. coli can grow in oxygen or without oxygen. And then every day, because they start to use up the glucose, the sugar, various nutrients, and build up toxic waste, somebody has to go in that lab every day and take about 1% of that culture and transfer it to fresh growth media containing some new glucose, which is the primary carbon source. Bacteria can utilize lots of different sources of carbon for fuel. They live in our guts. Most of them are very friendly, and they help us make many B vitamins, but every once in a while they'll acquire a plasmid, some auxiliary DNA that codes for a toxin, and you can get some really nasty E. coli infections that you hear about on the news. So some people think, oh, E. coli, that's scary. Most of them are great. There are a couple of nasty strains that produce toxins. The other thing that was in the buffer, it's very typical that you use a biological acid to buffer the media, and in this case they use citrate, and that's going to be an important point later on. So they estimate, or they calculated every day there's about 6.6 rounds of cell division or generations, and after 500 generations someone takes a sample from each of the 12 populations. And the cool thing about bacteria is you can freeze them. You stick them in like a 30% glycerol solution that keeps the ice crystals from slicing through the cells, and you can store them at minus 80 Celsius for many years. I think they have at least 6 freezers full of these bacterial samples in their laboratory. So as I just checked the website, and I guess it's on pause because of the pandemic. They had to freeze the cultures, and then maybe they're back to growing them again, but 76,290 generations since 1988. They did some competition tests where they take some of the frozen ancestral strains and mix them with the evolving strains to see if the new bacteria had developed any sort of competitive advantage and outcompete the ancestral strains. So, and then once the sequencing technology became more available and a lot less expensive, they started sequencing the genomes of the 12 evolving strains and compared them to the original frozen ancestor progenitor cells to monitor any genetic changes that were occurring. So what were some of the results? Well, for the first 20,000 generations, mean fitness increased 67% as the 12 E. coli populations became better adapted to the glucose medium. So their measurement of fitness was how quickly it can divide and grow in that medium. And we'll talk about why that is later. So basically what this means is the descendants were able to grow 1.67 times faster than the ancestor cells. Lenski says that every possible beneficial point mutation in the 4.6 million base pair genome of E. coli had already occurred many times over in just the first 20,000 generations. So a lot of the fitness mutations happened right off the bat. Many of them after just 5,000 generations. And then by 60,000 generations, the average fitness gain slowed down. So it's a peak that kind of eventually flattens. So just jumping from 20 to 60,000, you have a 3% increase compared to that 67% increase through just the first 20,000. Yeah, like Dan, I should have put a graph up there showing that. I have one later actually. So, and then after between 20 and 31,500 generations, something cool happened. Well, you saw this leveling off, but one of the populations had an accelerated growth rate. You see the flask was a lot more murky, turbid than the other flasks, which means either they had a contamination or one of these E. coli really seemed to adapt to its environment and take off. So this is exciting. Is this evolution in action here? Later on, they did some experiments, which we'll find out later, that there was a mutation that allowed them to take up citrate in oxic conditions. Normally, E. coli only takes up citrate in anaerobic conditions. Why was the citrate there? Because it's used as a pH buffer in the growth medium. So normally, the E. coli were only supposed to use glucose as their carbon source, but they found another way to get fuel from what was in the media. So yeah, E. coli normally only transport citrate under anaerobic conditions. They have all the enzymes for metabolizing citrate. Most cells do. It's the citric acid cycle. You might know it as the Krebs cycle. Citrate is just the ionized form of citric acid, and you find it in high concentrations in citrus fruits. So if you've ever had sour gummy bears, that crystalline stuff on the outside, maybe your salivary glands are pulsing right now just thinking about putting one of those in your mouth, but that's citric acid. So it's a totally fine acid. You can eat it, turn it into fuel. You can even turn it into amino acids as the metabolites and other wonderful things. So the gene for that is the CIT, the citrate transporter gene, which is under the control of a regulator that suppresses the gene whenever oxygen is present. So what did they find? There was a total of 110 genetic rearrangement events that were seen among the 12 cultures after 40,000 generations, as they reported in this paper. Found 82 deletions, including a couple of large deletions of up to 55 kilobase pairs were found in all 12 populations. And these were the most frequent type of rearrangement event. They had 19 inversions, 9 duplications, and the genome sizes were reduced by amounts ranging from 0.9% to 3.5% compared to the ancestral genome due to all of the deletion events. 6 of the 12 populations developed defective DNA repair systems and now acquire slightly deleterious mutations at a much higher rate. So every time the cell is dividing, there's quite a few error checking mechanisms, and if the E. coli gets a mutation in one of those, then a lot more mutations are introduced. So some of those cells now have over 1,000 mutations as a result of these defective DNA repair mechanisms. So it accelerated the mutation rate. Sanford did an experiment with a computer simulation studying bacterial genetic entropy, and they found that using the same growth conditions and mutation rates as Lenski, the cells by 60,000 generations would have acquired about double that. So double the number of mutations seen at 40,000 generations in the simulation as well as in Lenski's experiment. So in silico and in vitro. But even those cells, the populations with functioning DNA repair systems, they are still acquiring so-called bad mutations at a much faster rate than any of these fitness improving, these good mutations. So let's talk more about these citrate positive mutants. Normally, E. coli would be categorized as citrate negative, even though it has the ability to process citrate, it just doesn't import it unless you remove enough of the oxygen. Not all that much oxygen in our colon where they like to live. The new adaptation was caused by several complementary random mutations, which allowed unregulated or continuous expression of the gene that controls citrate uptake. So what happened is there was a duplication event, and it copied a gene regulatory region that's normally upstream of a gene, and it controls whether that gene is going to be turned on or turned off, transcribed and translated in a protein. And so this regulatory region was duplicated and ended up inserting near the citrate transporter gene. So instead of having this gene be suppressed by its normal regulatory suppressor system proteins, this one is now being constitutively expressed when oxygen is present. So it now acquired the ability to import citrate and use it in the citric acid cycle, even when oxygen was present. So this is still considered, we're going to talk more about gain of function, loss of function mutations, but in this case, what happened is there was a loss of gene regulation. The regulatory feature that suppresses the citrate transporter gene was lost or overridden by the now oxygen dependent activator. So
James, if I could just for a second, I just want to put a pin in that, because that's the way you describe that is not exactly what happened. It wasn't the promoter that got duplicated, it was the CYT gene itself, and it landed next to a different promoter. We'll come back to it. I just want to put a pin in that.
Good. Yeah, thanks for correcting that. When I was reading a summary of it, it was kind of confused about which of the genes got duplicated. But in any case, you end up with the citrate transporter gene being next to a promoter that allows it to be activated. And yes, if you, obviously I haven't gone through all 264 whatever papers, so if you guys catch any other errors, especially if it changes the outcome of what I'm saying, please do let me know. You can email me at chemistryofcreation@gmail.com or mention it to Dr. Dan. So a couple of other scientists have done a similar experiment where they repeated this long-term evolutionary experiment with E. coli. And the principal investigator was Scott Minich, who's he is a here's the paper: rapid evolution of citrate utilization by E. coli by direct selection requires these the citrate transporter and then another gene. So this is published. Didn't put the year. Here's the.
I have that one right here. That was 2016. I have, if we want to at some point have it, I have it right here if we want to.
I have the paper. Yeah, I guess I left out the date. Sorry. So yeah, Dr. Minich is the senior fellow at the Discovery Institute. So you can read more about him here. So these intelligent design scientists are doing research, doing publications here. So what did they find? Well, Lenski's research required over 30,000 generations and many years to get the citrate positive feature. But interestingly, in the repeated experiment, Van Hofwegen and Minich's team saw the same mutation come up after only about 12 generations and 30 days. I don't think it's the same exact places where I don't think it inserted in the same spot, but it had the same outcome. It had the effect of being able to
the same regions. It wasn't exactly the same sites, but it was the same regions. Yeah.
Right. So this suggests that the mutation really isn't that complicated or hard to get. Now, this is only one repeat of the experiment. Sure, if someone tried it again, maybe they wouldn't have gotten as lucky. It could have taken a longer period of time. And I need to go back and look at their material and methods and see if they exactly mimicked the conditions of Lenski, or they tried different selective pressures, like putting more citrate to begin with, things like that.
Yeah. They messed with the genetic background. So they tried a couple different genetic backgrounds, including ones that had what they were hypothesized, basically, I think they called them potentiating mutations. So they worked in a couple of different backgrounds that had the potential pre-existing mutations that facilitated the CIT plus trait, and they put them in an environment with citrate as one of the primary organic compounds available. So basically they imposed a selective pressure to basically say, if you can use this citrate, you're going to win, and gave them kind of the genetic background, where some earlier work that Lenski's team had done showed that, like, after, I forget, it was after like 20,000 or 25,000, there were a few lineages that were more likely to subsequently evolve.
Yeah, kind of like the precursors to the
exactly.
The ones
so
that they got
and then Minich and company, I think it was Van Hofwegen was the first author.
Van Hofwegen.
They basically used those genetic backgrounds and said, okay, what do we think those mutations are? Let's see if we can then get it to take that third step in effect.
So in essence, they kind of increase the selective pressure for getting the citrate mutation.
Exactly, yeah.
And fortunately, they didn't have to wait as many years to get the results. Okay, so correct me if I'm wrong, but this is a comment that I think you made 4 days ago on the Peaceful Science website. My favorite thing about this experiment, referring to the Lenski experiment, other than the simple fact that it exists, is that the aerobic citrate metabolism trait is a directly observed experimental refutation of irreducible complexity. So I imagine that's what we'll be talking about later. Multiple mutations required to have the trait, no selection for intermediate stages with a subset of those mutations. And one of the co-authors on a paper that showed this to be the case was Scott Minich. Is it Minich or Minich?
Honestly, I was going to ask you because I don't know.
I haven't met him yet. I just looked at his bio, and I couldn't tell by reading how to pronounce it. So my apologies if you're listening and I mispronounced.
Sorry,
Scott. So yeah, at the University of Idaho and the Discovery Institute. Oops. Let's. I got a couple of comments from their paper in regards to this. As in terms of the mutational pathway that was involved, it essentially involved breaking something.
Well, right off the bat, though, that's not true. It didn't break any repressors. They moved the CIT plus gene, but they didn't change anything about any of the regulation for either location. It copied from one place where it was under an anaerobic promoter, and then it moved into a region that was under control of an aerobic promoter. Both the promoters work the way they always have. There was no loss of any regulation for either of those sites.
Are you talking about in the Lenski experiment or in the second experiment with Minich?
As I understand it, the CIT duplication is the same in both cases.
Right. Well, I guess what they're saying is broken is the previous ability to suppress that gene under oxic conditions, which presumably has some sort of purpose for not transporting the citrate, because it's also, I think it's an antiporter. So as it imports citrate, it also exports succinate, fumarate, and and one other one, forgetting now. But anyway, you may not want to deplete those intermediates if just for the sake of getting an extra carbon source, unless you're desperate and starving. So maybe that's what they mean by breaking it, is interrupting the normal regulatory function of the repressor.
But again, you're not messing with the regulation of either of those. You have the original citrate module, right? The CIT-T module, where it's only expressed under anaerobic conditions. And then you have it gets duplicated upstream into the, what's the name of the gene? So RFK, something with an R.
I think I have it in the table. Yeah.
Yeah, it's over the next.
It's hard to remember those acronyms.
There's a gene upstream that has the promote, where that next module upstream is under an aerobic promoter. So when the duplicated CIT-T gene lands under control of that aerobic promoter, now you can still regulate that second copy. It's just now you can have it expressed under aerobic conditions. It doesn't have to be, because all the other stuff about that second copy, you're still regulating to whatever that module is. You're just now able to express CIT under aerobic conditions.
Yeah, maybe breaking isn't.
What I would ask is it look at the inverse. If it started with two CIT genes, one that was under aerobic expression regulation and the other under anaerobic, and it lost the module that allowed for aerobic CIT expression, we would all be agreeing, everybody would be agreeing that that's a loss of function. Mm-hmm. Right? You had two of them, you could express under one set of conditions, you could express under another set of conditions. And then if you lost one of those, we would all agree that that is a loss of function. Mm-hmm. Right? So then how could the inverse of that also be a loss of function?
It'd be more like an alteration of the wild type system, I guess.
Right.
So the other thing that was altered is as I understand, more of the citrate importer was produced. So that, I guess you're talking about the duplication of the transporter gene. So would that end up making more of the protein? Yeah, because you're complex, the membrane-bound transporter.
Yeah. Yep.
And then I guess there was also a succinate importer that I believe was duplicated.
I think that was CIT-A, and you're upregulating CIT-A and that was one of the mutations that actually made the CIT-T duplication beneficial, because as you said, if you're getting rid of is it I'm not a biochemist, is it succinate? How do you say that?
Succinate, yeah, succinate.
And
fumarate and
yeah. Well, if you're getting rid of that carbon source, right, and importing citrate, what's going to matter in terms of the balance of that is going to be how much of each of those things you have. So it's maybe beneficial, maybe harmful. And we know that under the Lenski baseline conditions, just duplicating the CIT-T, the antiporter, without any of the other mutations is strongly harmful, because people have gone through and done kind of one at a time experiments with each of these mutations. But if you pair that, so you're bringing in more citrate, but getting rid of other stuff, but you're pairing that with an increase in your ability to bring that other stuff back in, now you've removed the cost to that duplication. So upregulating that second transporter is one of the kind of background mutations.
Like a recycling mechanisms.
That was required.
We didn't want to send it out, but we got a way to bring it back in again.
Exactly. So now you're now you're getting an extra food source here, but on net, you're not losing this other one that you already had. So that's
sort of a little bit of a rescue mutation. It's kind of like, it turned out to be beneficial, right?
It's a compensatory mutation. Yeah.
Right. Okay, let me make sure I'm on this.
So yeah, so you need it's the CIT-A is the other transporter to let you re-import some of the stuff that you're getting rid of with the antiporter. Obviously, you need to duplicate CIT-T into the aerobic promoter region just upstream. And then the other thing it turns out you need, and this was a really cool finding, was there was a gene that was upregulated that had to do with acetate metabolism. And this was the other one of the major priming mutations that had to occur earlier, because this one, it had to, I don't know, again, not a biochemist. I don't understand the biochemistry of how these different things interact with each other, but by doing something to your acetate, by upregulating part of your acetate metabolism pathway, that made it easier. Oh, well, I guess it's because you're getting acetyl-CoA feeds into the citric acid cycle, I guess would be the reason why.
Yeah, so combine with oxaloacetate to make citrate.
Exactly. So if you're doing something with that, then that's going to increase,
help replenish the cycle.
Exactly. So that makes sense. I remember biochem. It was a long time ago, but I remember a little bit.
I know. So some of this stuff has been like 20 years. I mean, the biochem I teach all the time, but the stuff I learned about evolution was mostly undergraduate school more than 20 years ago.
So the cool thing about this acetate one is it's neutral with regard to the citrate stuff, but it's beneficial along a different dimension because you can increase the rate of acetate metabolism. So you upregulate that, and that doesn't have anything to do with citrate on its own. And if you have citrate on its own, that's bad, but you have the upregulated acetate metabolism plus the upregulated other transporter, the CIT-A transporter. Now you get that CIT-T duplication, and now you're off to the races because you can you can have a major fitness advantage with your ability to utilize citrate efficiently. And
What's
that's
interesting is it probably had to have occurred within the same population fairly quickly.
It was over the span of about 10,000 generations, I think. So when they went back and redid it, so the original Cit Plus mutant was somewhere in the low 30,000s, and they went back, and prior to 20,000 generations, it was very rare to be able to re-evolve that. But between 20, 25, 28, somewhere in that range, it became much easier to recapitulate that evolutionary pathway, showing that those mutations occurred somewhere in that range, and then basically just kind of puttered around due to genetic drift. They weren't selected for, but they also weren't selected against. They were just hanging out in that population. And then when the CIT duplication hit, that whole set of mutations as a unit became beneficial. And that's why in that comment you showed, I describe it as an observed example of an irreducibly complex trait. That system meets Behe's criteria as he described it in Darwin's Black Box. So like, that's like one of my favorite things about this experiment. Like, there you go. We have a can you evolve an irreducibly complex trait as Behe describes it? There, yes, we just did it.
The paper that I remember, they were showing how they had trouble getting it back at the 20,000 population. So it was somewhere in between the 20,000 and 31,500 or something like that.
Sorry, I just need to correct myself. It wasn't CIT-A, it was DUCT-A, DCT-A was the other one, not CIT-A. Sorry.
Right, yeah.
It was DCT-A, not CIT-A, sorry.
It was back a couple slides, you know.
Yeah.
Oh, well. Okay, so this was the response from, or this is what the authors stated in the paper: no new genetic information or novel gene function evolved. The LTEE has not substantiated evolution in the broader sense by generation of new genetic information. In other words, a gene with a new function. And then they said, because this adaptation did not generate any new genetic information and only required expanded expressions of two existing transporters, the CIT-T, and there we go, the DCT-A, generation of the E. coli citrate positive phenotypes, in our estimation, do not warrant consideration as a speciation event. I remember a couple years ago, there were some scientists were wanting to call these a new species of E. coli, and so there's some debate about whether that qualified or not.
Yeah, because one of the defining characteristics is that E. coli cannot do, it's a diagnostic test for E. coli is, can it do aerobic citrate metabolism? And if the answer is yes, then it's not E. coli. So
right. We do that. I taught microbiology years ago, and we used to do that test to see if they, they had to try to identify the unknown, and then they would do all these different
I remember
chemical tests and biochemical tests. Oh, this one didn't grow on the citrate positive plates. And my wife's her PhD is in microbiology too, so she's teaching the micro. But so usually that's how they would identify at least a new strain if they didn't call it a new species, whether if it had lost or gained a different metabolic ability. So in terms of, Dr. Dan already explained some of this. So why did the citrate positive phenotype identified by Van Hofwegen et al. Appear so quickly, whereas Lenski's research took years? Well, part of that is because they seem to set up the experiment to be more have heavier selective pressures for it. So there was a commentary article by John Roth and Sophie Maisnier Patton. I'm sorry, I can't pronounce her name, but they said the primary message of the paper is that the series of events used to explain adaptation in the short transfer LTEE and in speciation might need to be revised. It would appear that the delay in the LTEE experiments may not reflect need for a neutral potentiation step, but the difficulty of intermittent selection to act on frequent copy number variants. The bottleneck is in serial dilutions is hard to cross when initial improvements are due to an unstable copy number variant that is counter-selected during the intervening rapid growth period. That's a big, complicated paragraph there.
Basically, was saying if you impose strong genetic drift, it's hard for rare intermediate mutations to survive. Which,
exactly,
absolutely. you're likely to lose rare variants when you go through a bottleneck like the day-to-day transfer between flasks. Yeah, absolutely. And I think that's exactly the explanation for why it took so long in the Lenski experiment, but then they were able to do it relatively quickly by setting it up and imposing conditions that make it easier to select for those states and reducing the likelihood that you get rid of them accidentally through drift. Then you're gonna arrive at the combination more rapidly. I think that makes perfect sense. I mean, I disagree with their conclusions. I read this paper, and it's funny because they go out of their way a bunch of times to be like, here's this experiment we did. We showed that the Lenski experiment did it's like hardcore experimental biochem and evolution interspersed with, and by the way, this disproves evolution. Like the way they write the paper is really funny, that there's like just a couple little lines like, by the way, this isn't new. It doesn't actually count. And I find that funny because one, they use the phrase, there's no, new gene function. Well, there is a new gene function. There's the CYT transporter is expressed under aerobic conditions. That is a brand new function that E. coli don't have. But the other side of it, and the reason I really adore this paper, is because it shows exactly what like people like me say in response to like he's saying these complex traits can't evolve. You can't get new, systems that require lots of components that interact with each other. And this paper is a really beautiful illustration of exactly how that process works by showing that under certain conditions, you can actually get these types of systems really easily. And you don't need to like poof new genes you don't need to just get a de novo gene out of nowhere to get a new trait. You just need to kind of rearrange things. It's tinkering and tweaking and rearranging, and you get some pretty significant new traits. And it doesn't stop with just the citrate metabolism, because what you see here is you start an environment where it's just glucose is your food source. And then one lineage, one of these 12 cultures gets the ability to use citrate. Well, that has a bunch of downstream effects because now that's growing to a higher density. And there was a paper just last year in 2020 that all the creationist organizations just glommed onto, because they showed that in the original culture, in the one of the 12, that was I think it was ARA -3 was, I forget which one it was one of them. One of the 12, they have higher mortality in there among the CYT plus culture compared to the other 11. And if you take it out of the normal growth medium and you put it in citrate only, it has really high mortality. Well, for the second one first, of course, it's going to have higher mortality in the citrate only environment because it didn't adapt to that environment. It's not right. It's been selected for one environment, and you put it in another one, it's going to do worse. That's like, nobody should be surprised by that. But the other one is in the original environment. It has higher mortality. Of course they do. They're grown to a much higher density. So you get much higher competition, which means you're going to get more mortality. That's just that's exactly what you'd expect based on the ecology there. And it actually led to a really interesting situation where you see subsequent adaptations for metabolic pathways involving lipids. And it turned out what they're actually doing is using they're using the dead cells as a food source. And so what we're actually seeing here is a multi-step progression of basically ecological succession in this single flask, where it's essentially niche creation, or niche construction is the word for it, where they're by getting one new trait, aerobic citrate metabolism, that allows you to grow to a greater density. And that greater density imposes stiffer competition because eventually they still run out of food. So in this higher density population, a lot of cells die because that's just the nature of how it works when you have a limited food source. But then the survivors are now under selection to utilize those dead cells as a food source. And we see those subsequent adaptations in that same population. So not only do we see at the biochemical level how you can have a complex trait evolving, right? And Minich and other people showed it requires a number of mutations, any one of which on its own is at best neutral, several of which are actually strong. The CYT duplication on its own is strongly harmful for the reasons you explained. But then you could even go up a level from that to the ecological level, right? And see how you can have niche construction, and by getting a new biochemical trait, that leads downstream to ecological changes in that population that now give you another new resource, and that changes the selective pressures to adapt to utilizing that new resource. So you see this really nice domino effect of adaptation, open up a new niche, leads to an ecological change, opens up another new niche, and now you adapt to that. It's a really cool series of changes in that CIT plus line. And again, like I said in that comment, it starts with the evolution of an irreducibly complex trait.
Well, we'll need to come back and describing more about what a functional system is versus a functional protein and what irreducible complexity is. That's probably going to take another lecture to talk about that. Let me just finish these last few slides and then we can open that up for discussion. Yeah, so more I'll just finish the comments that they made. We conclude that the rarity of the LTE mutant was an artifact of the experimental conditions and not a unique evolutionary event. Again, you're creating, like you said, a micro environment where the bacteria is saying, hey, all we're given is one thing to eat, there's glucose here. And if there's any way that a mutation can allow us to utilize something else like citrate or even the dead remains of our of our surrounding cells, anything to stay alive and get a new way of utilizing resources. But to do that wasn't necessarily the addition of novel genetic information or no new genetic information. And I'm sure you're familiar with Casey Luskin. He emailed me or a quote that he had about, he said, when you impose very strong selection for growth on citrate, every step yields a functional advantage. It's not a neutral pathway, and Lenski's story isn't one of neutral evolution evolving a complex feature, but rather one where each step gave a successive advantage, and no step creates something new. So
but that's just not true, though. Because if each step created a successive advantage, we wouldn't have seen that fitness plateau followed by a subsequent increase. We would have seen a smooth rise as those earlier mutations occurred. The reason they had to go back and figure out what the genetic background was for that trait is that you had a sudden, it had more or less maxed out at about a relative fitness of 1.6, 1.7. And then the CIT plus line restarted that trajectory and the other 11 didn't. If each step in that pathway conferred an advantage, it would have been a smooth rise to its ultimate. I forget what the ultimate relative fitness is for the CIT plus line, but it would have been a continued smooth increase to that point, not the pause and then the restart. But going back through the earlier generations, we know that those mutations were there 10, 12,000 generations prior to the CIT-T duplication. So, I mean, he's just wrong when he says that each step gave a successive advantage. That's not true. There's no benefit to the DCTA transporter on its own. There's some substitutions to CIT-T that were neutral on, like single base substitutions within CIT-T that are neutral on their own. The duplication on its own is strongly deleterious because you're basically wasting energy using a food source at the expense of more common sources of carbon. So, I mean, he's not correct. Like, he's just factually wrong when he says that there's a successive increase with each step. It's not true. And we could go back to the original Lenski, like, you don't even have to go into the genome. You can just look at the fitness trajectory and see that that's not the case.
I'll double check with him, but I think when we have to distinguish whether he's talking about each step in terms of a mutational event or each step in terms of a selective beneficial advantage, where it gets more locked in because of either shedding excess baggage of the genome for things that are no longer needed, like these E. coli had mutations in the flagellum to begin with, so when it started to get rid of some of those genes, it made it able to reproduce faster. So are we talking about a step in terms of a gene alteration event, or a step in terms of a successive advantage, like you're talking about, where there's some kind of selective advantage that maintain that population into the next round?
I think in this quote, he's specifically talking about citrate because he says when you impose very strong selection for growth on citrate, every step yields a functional advantage. But that's not true. Some of those intermediate steps are neutral, and some of them, like if you just treat the simplest version of this is the acetate mutation. I forget what that one does specifically, and then you've got the DCTA, the transporter, upregulate that, and then the CIT plus antiporter duplication. That's like the simplest version of this. The two of those, the single base substitutions, the acetate one and the other upregulating the other transporter, those are both neutral on their own. And the CIT-T duplication is extremely bad on its own for reasons you explained earlier. So when he says each step yields a functional advantage, it just doesn't. Like, that's not what the data show. And we've directly measured.
I'll ask him to clarify that because, yeah, I see your point. It does seem a little confusing the way it's worded there. I guess the editors also gave a response from the Journal of Bacteriology where this is published. They said that in the issue, the author suggests that the long-term evolutionary experiments of Lenski and coworkers may need to be reinterpreted. I'm not sure if they need to elaborate on that more, which specific component of the experiment they're referring to, that was just the citrate by itself or the other mutations that gave a fitness advantage by shedding some of the unused genes since they were in a very simple growth medium.
Right.
And then the other analysis was done by Christopher Rupe and John Sanford. They have where did I put the paper link? Oh, I guess I gave it in the beginning of the slide. Yeah, like along with what you were saying, there's at least in some of these older papers before they even identified what the mutation, what caused the citrate to be able to intake more E. coli, but they hailed it as a key evolutionary innovation. Lenski did, and then the New Scientists claimed that this is the first time evolution has been caught in the act of making such a rare and complex trait. And Sanford and Rupe said that, so this is their quote, the bacteria have not experienced forward evolution, but rather the net effect has been reductive evolution, or evolution going backwards, according to them. They said in all 12 experimental populations, the functional bacterial genome has shrunk, so it contained less total information. Initially, about 1 in a million mutations was beneficial in that particular selective environment. Obviously, you take them out of there and have them compete with a wild type, then it's probably no longer beneficial. But by the time the total population reached 100 trillion cells, there were a combined 100 billion mutations, with less than 100 of those mutations considered beneficial, having some sort of functional benefit in that environment. So 1 in a billion mutations. So only one of those mutations could be considered a gain in function mutation, which was the constitutive import of citrate in oxic conditions. So that superficial fine-tuning has been happening at just a handful of genomic sites. Significant genetic damage has been accumulating through the rest of the genome due to many slightly harmful deleterious mutations that cannot be selected away. This means that in the long run, the net effect will be degeneration. Sorry, what?
Can we pause right there? Once you finish this, now I just want to pause on that because that's one of Sanford's like catchphrases, and I want to ask about that.
Sure. You want to come back to it after, or right?
Well, I just want, if you don't mind, I want to just ask like
what am I on? 25. Yeah, I got maybe another 15 slides, but he's referring to just how when you get a alteration in the gene and it doesn't confer a benefit, but neither is it a lethal mutation, so it's carried on, that they start to accumulate because there isn't enough of a evolutionary selective pressure to get rid of all of them, since they're not lethal, and they're not immediately beneficial, or they're neutral or near neutral is what they like to say. So that's their whole principle of the genetic entropy
right?
of the genome, right?
And that's the other reason I love this experiment, because it is a direct experimental refutation. In addition to of irreducible complexity, it's a direct experimental refutation of genetic entropy. Because here we have Sanford characterizing these mutations, and the genome shrink, and you lose functionality, right? And you only have 100 beneficial mutations, and all these harmful mutations can't be selected against. Putting aside how he is characterizing that they are deleterious, because I don't think Sanford has gone in and characterized most of these mutations, or any of them. Putting aside how we're characterizing things as slightly deleterious, if Sanford is right, then the fitness should be going down. But what we see is that the fitness goes up across the 12 lines by significant margin. So it's not just 1 in a million shots like the CIT, like the CIT plus trait that allows it to increase. It's just the things they're doing are beneficial in the context of this experiment. Now, you're right that some of these things are going to be harmful if you were to take them and put them back into the wild. They're going to be outcompeted by the wild type E. coli. But that observation itself is fatal to the idea of genetic entropy, because inherent to that idea is the idea that that mutations have a fixed fitness cost independent of context. That if you have a population that experiences every possible mutation, on net, the cost of those mutations will be negative. And that population will therefore be in a terminal decline, and no degree of selection is going to be able to rescue it. Now, if we're going to turn around and say, yeah, but bacteria, specific conditions, big population sizes, low mutation rates, whatever we want to say, a bunch of Lenski's lines actually increased their mutation rates. And one became a mutator phenotype, which really ramped up its mutation rate. And that one actually has higher fitness too. So that would seem to cut against what Sanford is saying. But more importantly, once you allow context specificity to enter the discussion, once the fitness effects of mutations are no longer inherent, and you can't just look at a genome and say, on net, deviation from this genome will be negative because it's based on this environment or that environment, genetic entropy no longer applies, because now you can always come up with a situation where selection is going to preserve some subset of that population, unless you want to posit that every member of the population experiences an exactly equal change in relative fitness each generation, which I don't think anyone is suggesting.
Well, the only criteria they're looking for at fitness is how quickly it reproduces. So that can't go on indefinitely. I mean, eventually you reach a peak, and in fact, like I think you were alluding to in Behe's 2020 criticism, that the death rate was rapidly climbing. Some of the populations were approaching a 50% death rate, and it was like 40% just a couple of years ago and 30% before that. So eventually you start to see this degradation. But just the fact that all of the population's genomes are shrinking, I mean, that is a loss of information. How can that not be genetic entropy if those genes were there? And unless you assume that those genes were junk DNA and they just got rid of the junk, I mean, eventually at some point you're going to start, you're getting rid of things that would be beneficial in an environment, different environments. So
you summarize my point to a T. Eventually,
so what you're saying is if you put these in the environment, then they would retain those genes. You wouldn't see the drop in population. I mean, in the genome size.
If you put them back out into the normal environment, they would probably go extinct. They'd be outcompeted by the E. coli that are out there. But if Sanford is correct about genetic entropy, then the loss of information that we're seeing in the Lenski E. coli genomes should be happening in all genomes. It shouldn't just be ones that are in a specific environment in a lab where you don't need 0.5 to 3 whatever the numbers are percent of your genome, right? The reason, so that's one. if Sanford describes this in like he's very particular in describing it as a universal process. So if this is a thing that's going to happen, then it shouldn't just be in this hyper-specific experimental environment. But two, we shouldn't be able to point to things like the fact that they lost certain genes as an example of genetic entropy, because we could show that there's selection in favor of those losses. The point of genetic entropy is that you have changes that have non-detectable fitness effects, but that still carry a cost in terms of being able to survive and reproduce and compete in some way, right? That's the inherent tension there, is that these traits that we're talking about, these losses that creationists point to and say, aha, genetic entropy, there's selection going on there. The point of genetic entropy is that there can't be selection against the losses that are accumulating. The fact that there's selection for them means that there could also be selection against them, right, in a different environment. And that means that it's not genetic entropy that's running the show here.
I just thought of a cool experiment that someone could do. Off the coast of California, it's really cold, and I guess found because of the sewage, doesn't kill every one of the bacteria that goes through our sewer systems. And some of these went down where it's really cold at the bottom. And because of the flow of water that brings cold water up to the surface, they were able to find some antibiotic resistant bacteria against a antibiotic that hadn't been used in like decades or something. So it would be interesting if we could find, or even like, take one of a frozen mummy or something and culture, see if we could extract some of the DNA from ancestral E. coli and see how much it changes to a sampling of E. coli that are present in human guts throughout the world. Just see how much, if there has been that kind of a genetic entropy, genetic degradation over the decades, because then you would have a wild type. You'd have a natural environment for which some of those genes could be selected for or lost depending on their the circumstances. But
I want to be very clear for the audience, just so everyone's on the same page. There's no such thing as genetic entropy. It doesn't exist. It's the math of the population genetics for genetic entropy. Like, it doesn't work because it requires completely unrealistic assumptions. The assumptions being that each fitness effect needs to have a fixed, or each mutation needs to have a fixed fitness cost, and it can't be context specific. You need to have, at no point can you reach an equilibrium between beneficial and harmful mutations, even as harmful mutations accumulate, which is in contradiction with the fixed fitness cost aspect to it, because if you flip something from good to bad, now it can flip back from bad to good, and now you've changed the relative frequencies. But you need a linear accumulation. It also assumes that the population cannot experience selection, even if, as you kind of described earlier, you get to a point where in whatever environment it's in, you're going to have a problem where it loses too many genes or, breaks a pathway that it actually needs or something. The assumption is that that cannot be selected against under any circumstances because the whole population is experiencing this degradation. But the problem is that there's always going to be differences in relative fitness within the population. Unless everybody is experiencing exactly the same decline at exactly the same rate, you're going to be able to pick, like, selection will just preserve the ones that are working, and they won't, and you'll get rid of the ones that lost the important things that are like, I would love to, like, I have a copy of Sanford's book that I've written in the margins. I've highlighted the crap out of this thing, and like, he's making a population genetics argument, and there is no math in that book. And like, I would love for somebody to actually do the math on genetic entropy and show like how it's supposed to work, because like in my PhD work, I used a mutagen to try to impose error catastrophe on viruses, and like, you should be able to do that. You can do the math. I did the math and showed like, I am imposing a mutation rate at this rate per genome per generation, blah 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 blah. I should be killing these viruses with mutations. Couldn't show that I was actually doing it in practice. Like, couldn't actually show it.
Now, when you say there's no genetic entropy, I mean, wouldn't you say even in this artificial environment, when all of the populations are losing genome size, wouldn't that be genetic entropy? I mean, even if you want to isolate it to just artificial environments, that is a loss of information. I don't see how that wouldn't be a genetic entropy. And then
Two reasons. There's
go
two
ahead.
reasons why it's not. One is that their fitness is increasing, and the genetic entropy hypothesis requires that fitness declines over time. And two is that the changes that we see in the bacteria in the Lenski experiment are due to selection, whereas genetic entropy requires that they are small, the effects are small enough that they cannot be selected against. What we're seeing in the Lenski experiment is that the changes are actually being selected for. So for those two reasons, it's not genetic entropy.
Well, you're only talking about the genes that were selected for, but throughout the rest of the genome, there's been accumulation of mutations as well that are not selected for, which would scramble the code, scramble the original genetic template, the genome. And then had another point about.
I would just say on that point, because that's what Sanford said in the quote earlier, he hasn't done the work to actually check to see if those mutations have any biochemical consequences for any of the genes that they occur in. And we know that synonymous substitutions are more frequent than non-synonymous. And even among non-synonymous changes, if you take two amino acids that are similar enough, there's no detectable difference in like the rate at which that enzyme is going to work. Plus, you can have huge differences in protein structure as long as the active sites and the overall structure is the same. So you can have two proteins that do approximately the same thing, but have like maybe 30 or 40% amino acid identity between them. So it's on Sanford and other creationists who are claiming that these mutations that have accumulated in the E. coli genomes here, it's on people making the claim that these are actually unselectable yet also deleterious mutations, which by the way on its own is a contradiction in terms, because deleterious means hurts your reproductive output, and that by definition is selected against. But putting that contradiction aside, it's on the people claiming they are both deleterious and unselectable to first show that they are actually deleterious in some quantitative way. And just showing that the mutations occur and they scramble the code, that's not going to cut it. You need to actually show like that those mutations have some kind of effect on like there needs to be, in order to be on the pathway to extinction, they need to have a detectable effect on some physiological characteristics of the cells. And if we're putting aside the ones that they're selected for that reduce the genome size, putting those aside, you've got a whole other set of mutations that are just there, but they don't we can't tell what they do. We don't know what they do if they do anything. For some of those, maybe they have an effect in the context of other mutations, right? As we saw with the CIT plus, because prior to the CIT-T duplication, those other mutations, they would just be scrambling the code. No apparent benefit, no apparent cost. They just scramble the code, but then you get that duplication, and now all of a sudden you have this new beneficial trait. So it's on creationists to actually do the work and like demonstrate that their claims related to the effects of this mutation accumulation are actually what they say.
Sure, there's a lot of potential fruitful research there in trying to determine if even if it's just a point mutation, if this has some sort of effect on the fitness of the cell. And in some cases, like you said, if you change one non-polar amino acid to another non-polar, may not affect the protein that much, but it might have a subtle effect that may not be perceptible until either down the road or when certain environmental conditions change, which is really difficult because then you've got to do all the different permutations of, okay, instead of just glucose, we're going to add maltose, or we're going to change the temperature a little bit, or we're going to, there's thousands of different permutations that you could do to try to figure out, wow, this accumulation of errors that didn't seem to be immediately beneficial or lethal, or it didn't have any kind of selective pressure in the current environment, does not mean that it wouldn't be selective in a different environment that we just haven't figured out. And sometimes it might take two mutations. Like what if you mutate a non-polar amino acid into, say, one with a cationic side chain, and there's another chance for it to bind to an anion, and it's not until you get another mutation that now causes the positive and the negative charge to attract each other, and that misfolds a different part of the protein. So it took 2 or 3 mutations before it became. So it's still, yeah. I mean, I guess you could still I would still consider that a entropy, because it's part of the pathway of leading to potential misfolded proteins and dysfunction in the cell.
So 2 things with that, and I agree with all of what you just said about how you can have downstream consequences of at first neutral changes. The problem is, 1, that is contrary to Sanford's genetic entropy argument. He's not talking about that stuff. He's saying that taken on its own, the vast majority of substitutions on their own have a small but non-selectable cost. Independent,
slightly deleterious.
Right. Independent of downstream consequences, there's some, so we're not talking about context specificity. You can make a different argument, but it's not Sanford's genetic entropy argument. But the second thing is that we actually have a really good example in what we've been talking about of what you just described, and it's that acetate mutation. And the paper that's relevant here, and I'm gonna, by the way, if you have any links that you want, like the Sanford and Rupe paper, stick it in the private chat on the side here, and I'll update the description of the video with the sources once we're done.
Oh yeah, good idea.
Oh, and I should say it's 10 after 4. We've been going for an hour, so like say another 10 minutes, and then we'll call it good.
Sure. Mm-hmm. I know you got to pick up your kid. I got to go get my son from school fairly soon too.
So let's see, it was the acetate mutation that has a really interesting history, because I simplified it before when I was talking about this. There was an original mutation in this acetate metabolism pathway that facilitated the CYT plus trait. It basically made the duplication. It made it from very bad to beneficial. But the interesting thing was, once you add on some of those other mutations that then subsequently, I forget the word they used, something actualization, optimization, whatever it was. There was like a 3-word pathway that they described for how this, like potentiation, actualization, something. I forget the 3rd word. Maybe I don't think it was optimization, but the 3rd word to make it better. And when you have subsequent mutations related to citrate metabolism, the interesting thing is that original acetate mutant becomes harmful in that new genetic context. So you actually see this cool stepwise trajectory in the fitness where it goes up a lot when you get the original duplication, and then you get subsequent mutations that actually cause it to go down a little bit. But then you find other mutations within the acetate pathway that actually change those dynamics and make it better again. So you get this weird staircase dynamic. And it's because of exactly what you just described, where you have a mutation. It actually starts out neutral. The original acetate pathway mutation is neutral. And then you get the CYT duplication and it becomes beneficial. And then you get subsequent kind of refinement of that citrate pathway. And that original acetate metabolism mutation is harmful, but you see other mutations that are then selected for in that pathway that recover it. You get compensatory mutations that actually bring you up even more. So so what you're describing is not a problem for evolution, because I think it comes down to it's in parallel nature, right? We're not talking about small populations here. So you've got lots of citrate metabolizing E. coli that are trying out, I'm personifying it, they're not consciously doing it, but they're trying out all these different combinations of mutations, and you end up finding one that turns out to be good, but then later on, now we've got new mutations and it's bad, but now you find additional mutations that make it that undo that harmful effect and make it on net good again. And the fact that we could observe something like that, like according to Sanford, that kind of thing shouldn't be possible, because the net effect of all these changes should inevitably be negative.
But again,
and I will say, let me just say, I'm gonna leave it there and say whatever you want to respond to that. And then if you want to take a couple minutes, just kind of wrap it up, and then we'll call it good.
Yeah, sounds good. I was just going to point out, and actually it's coming up in a few slides, that the fitness we're referring to is just one isolated type of fitness. It's just how fast can the cell divide. But when you're looking in the complete fitness picture, of course, we'll look at some of the other mutations that offered a benefit, but were the result of causing pre-existing functions to cease their function, and that offered it helped the one fitness aspect that they're looking for to increase the reproductive rate. Obviously, you can't, if you're going from cell division every hour, you can't get a cell division in 1 minute. There's only so much you can trim from the genome before you start running into damaging crucial proteins. So there would be a limit to that fitness test. But
that is the definition of fitness. That's
right. It would of that particular fitness test, which is how fast can these divide. But if we were doing another fitness test,
I know I said I do want to let you have the last word here, but I do need to say, that is the definition of fitness in evolution. It's not, there's not
being able to replicate.
Right. It's reproductive success. It's, how many kids and grandkids and great grandkids do you have? That, that's the definition. And I know there's arguments to change that to some biochemical engineering, whatever version of that, but at the end of the day, it's going to boil down to, are you able to survive and compete or not? The definition of fitness is what it is.
So instead of fitness, total functionality or something like that, capabilities, something like to that aspect. Where did I get? I think we're still talking about Sanford. He said the 3 questions that the experiment hoped to answer: would there be a net gain or a net loss of total biological functionality, I guess, instead of fitness? If we're just looking at the strict definition of fitness, which would be whether it can replicate or not. And then would there be a net gain or net loss of genetic information? Again, that's a whole nother can of worms that we probably don't have time to go down about what is information and what is code. Would a significantly new form of life emerge? Would there be macroevolution? Could we call it a new species? They say that there's consistently been a net loss of total function, a net loss of information, and the E. coli bacteria clearly remain E. coli bacteria. A new life form certainly not emerged. Now, some people will debate whether, because the definition of what a species is a little blurry. So at best, the genetic changes that have been observed reflect fine-tuning of a few pre-existing genes, or what is classically called microevolution. More fundamentally, in the bigger picture, there's been genetic degeneration, which is the consequence of increasing genetic entropy. Of course, he likes to throw in that term to bring back to his general theory. Lenski's method of measuring fitness was to test a single trait, which is growth rate, in a single environment, artificial bacterial cultures and shaker flasks, so they could be swirled around. They didn't have to swim on their own, on an artificial medium. In this context, Lenski's populations have only increased fitness in a narrow and artificial sense. However, in the fuller meaning of fitness, so this is where we probably need to change the term to just total functionality. All 12 of the populations have experienced a net loss of function, and so they have reduced total functionality as needed in the real world. And so they were showing as looking at the generations on the x-axis and mean fitness on the y, which is just by that single trait of whether it reproduces more quickly or not. Most of the beneficial mutations for that particular selection happened early on, but at the same time, they got rid of some of the other enzymes that use, for instance, to break down maltose or other sugars. They had mutations that occurred in some of these genes that were not needed because there was plenty of glucose around. They didn't have to rely on other substrates for catabolic, for turning that into fuel. So one particular trait gets selected for, but overall there's a loss of mean total fitness because of the mutations and those other non-used genes. So he said, in addition to the very rare reductive mutations that have enabled somewhat faster growth rates, there are a host of nearly harmless mutations accumulating in all parts of the bacterial genome, which have no benefit at all yet escape purifying selection and elimination from the population because they have such small effects. So they put this table of the various gene or regions that were mutated, including the function, which population number of the 12, how soon it was established. So most of them happened quite early on in the experiment, and how much of a fitness gain in terms of how their cell division time increased, their generation time increased. Some of them had quite a dramatic effect. But when you look at each of these mutations, you see either a reduction in the original function and a complete inactivation of the gene, and only the citrate positive one could be considered a gain of function feature, which still involved a loss of regulation of that of that gene that it is normally under the suppression of. The ability to uptake citrate was a result of the random duplication or copying of the already existing citrate transporter gene and its reinsertion or pasting into a new gene region near the R and K promoter that activated it. The citrate transporter protein could then be continuously expressed. So why did the cultures grow faster? Why was fitness increased? Because they lost non-essential functions in their artificial environment, such as the SPOT mutation that occurred in 8 out of the 12 populations. That does several functions, but one of them had control. It lowered the expression of the flagella encoding operons. And since these were non-motile strains to begin with, and the flasks were swirled, no sense in making a flagellum because it costs the cell energy. So just that mutation alone gave a 9.5% improvement in fitness, allowed the cells to do their job of being able to survive, grow, and reproduce without having to waste time and energy making a flagellum or components of it. In another experiment, scientists.
Hey, James, I don't want to interrupt you, but I do need to jet in just a couple of minutes. So if you want to,
I got like 6 slides left, I think.
If you can go super fast.
Sure. Okay. So they found that the flagellum uses up 4.5% of the cell's total energy in Salmonella strain, and skip that. So mutations that increase fitness, increase the growth rates, were also seen in genes involved in the catabolism of sugars other than glucose that were not present in the growth medium, such as ribose and maltose. So by getting rid of at least mutating enough so that you didn't have ribose or maltose, that increased, it got rid of the excess baggage, in other words. So Lenski acknowledges the mutations improve fitness on glucose while adversely affecting performance on other substrates. Researchers involved in a long-term experiment have misleadingly called this type of change adaptive evolution. In reality, this is nothing more than functional degeneration leading to habitat over-specialization. And Sanford says this only leads to evolutionary dead ends. And then really briefly, Michael Behe, in that paper you're talking about in 2020, said those initial random beneficial citrate mutations that had been seized on by natural selection tens of thousands of generations earlier had led to a death spiral. So the death rate of the ancestor was 10%. After 33,000 generations, it was 30%. And then by 50,000 generations, 40%. And now some of them are exceeding 50% in some of the later experiments. So no matter whether an organism is in a lab or in the wild, no matter which kind of organism, whether microbe, plant, or animal, it's subject to Darwinian mechanisms' tender care. So thanks to the Lenski group, we know that devolution is relentless. It never rests. In good times and bad, if a change in a species could help it adapt more closely to its environment, degradative mutations will arrive most quickly by far to offer their assistance. And of course, under the selective pressure, a species has no choice but to accept helpful ones, even if that eventually leads to the species languishing. Even if there are several possible pathways by which to construct a gain of function mutation or several possible kinds of adaptive gain of function features, the rate of appearance of an adaptive mutation that would arise from the diminishment or elimination of an activity of a protein is expected to be 100 to 1,000 times the rate of appearance of adaptive mutation that requires specific changes to a gene. So just to be able to get that to the point where you can have that function, the citrate positive gain of function mutation, there was 100, empirically and predicted to be 100 to 1,000 times as many of adaptive mutations that are that result in the loss of a function in order to speed up the cell division. Okay, so yeah, I'll just skip. That's kind of a repeat of what was already said. So yeah, I guess the point that Behe's making is, if you end up getting so many more of a loss of function, we saw the loss of ability to process ribose and some other sugars and all those mutations, it does make it seem like these losses accumulate at a much faster rate than any gain of function mutations, leading to an overall scrambling of the original genetic information. So we talked about the difference between artificial and wild type environment, and I think that's a good place to stop, because we can see if we can propose or look at any experiments where they've done this in differing laboratory environments, or comparing ancient versus modern genomes of cultures that currently live in the environment, including the human gut.
All right. Thank you, Dr. James Carter. That was, I had a fun conversation. I hope that was enjoyable for you. I hope.
Yeah, sorry, I've been so sleepy. I had a rough week, and I'm trying to stay focused on what you're telling me. I learned some new things too, and I got to go back and double check. So that's what I wanted to do. I wanted to lay some stuff out there, get some constructive feedback, and so I thought that was very fruitful. I'm sure it will open up a bunch of other interesting discussions later on.
Absolutely. I'd love to have you back on to talk more about this stuff. Either Lenski specifically, genetic entropy, anything in more general. We'll chat. Maybe we could do this again down the road. So,
sure.
Everyone watching, thank you for watching. Let's see. Do I have any notes for everybody that I want to just? Let's see. I know. I know Sal is having an after show. That's Evidence and Reason. So if you want to head over, if you all know Sal Cordova, if you want to head over to his show, Answers in Atheism, that's formerly Speed of Sound of Gravity, is doing his weather report. So if you all want to head over there to that show, I recommend doing either or both of those things. And last note is just, if anybody else is interested in this kind of conversation, I have a generally open door policy. So just email me. I'll put my email down in the description. It's creationmythschannel@gmail.com. And also, James, if you want to either just stick it in the private chat here, or email me with the sources you have, and I'll stick all those links in the description as well.
Sounds good.
Right. Thank you for watching, everybody. I will see you, I don't know, at some point. I'll see you down the road. Bye, everyone.